The Pipsqueak.
Sorry.
Welcome to the Pipsqueak Pro Webinar. Thanks for coming and joining us.
This is cell counting software.
Oh, cool.
So you
I
can
feel like, what is the Zoom now?
That's a great intro, actually. That's perfect.
Then you should do that on purpose.
This is cell counting software, yeah. We'll just give it another second here to let everyone else come and join us. And then we'll get underway. Generally today, I'm going to walk through some of the new features that we have in Pipsqueak Pro and give you a link to download it and try it yourselves, and answer any questions. I'll try to make this pretty to the point today. I don't want to eat up your whole hour. So we'll get through things here pretty quickly and then answer any questions that you have. Yeah, so Christy just popped a poll up there. If you have a second, go ahead and answer that. Looks like we got a number of new people here. That's awesome. So welcome to Pipsqueak. Welcome to Rewire. We'll talk a little bit about where Pipsqueak came from and how we got to where we are today.
Actually, a lot of people that haven't used it before. That's great. I'm excited to show it off. Okay, I'm going to switch to one more quick poll in just a sec. If anyone else wants to still answer this one.
I'm curious, for those of you who haven't used Pipsqueak before, what do you use currently to do your image analysis or cell counting? Feel free to hop on camera or pop that into the chat, but I'd love to know what you're using.
Hi. Sorry, can you
Hey, yeah.
see me?
Okay. Yeah.
Hello, I'm Anaya Kolic. I work on faculty of biology at University of Belgrade in Serbia. And I use, for my analysis of neural nets, I use so far ImageJ, of course, and well, I just do region of interest and like that. But I'm really interested in your program and your approach. It was
thank you.
To hear it.
Thank you, and thanks for staying up late. It's got to be quite early in the morning.
It's like 8.
Just... Oh, okay, all right. Well, thank you. That's great. Yeah. I'll talk a little bit about like the origins of this, but ImageJ is certainly where I was doing a lot of my cell counting during my postdoctoral fellowship, and so Pipsqueak was one of those great ways that we figured out how to automate this process and speed it up. So nice to see it benefiting others as well.
Yes, it's amazing. I guess you use some kind of program when you detect edges of high and lower contrast pixels or something like that.
Yeah, that's actually a really good segue into what we'll talk about. Yeah, the very earliest versions of Pipsqueak were using thresholding entirely, and that had some benefits to it in that it's good at picking up a lot of different cells and stuff. But if you have high backgrounds or a lot of like white fibers going through your image, it can really fail at finding edges. And so that's where our AI approach came from, and I'll talk about that. Christy, did you have one more poll there?
Not right now. So
okay.
Go ahead once we get started.
Cool. All right. Well, yeah. So a little roadmap of what we'll talk about today, a little background on where this has come from, and then I'll show you how to get going with Pipsqueak Pro. We've actually got the beta release link available for you today. And then I'll talk a little bit about where it's going to go from here and what you should be watching for. And finally, how to navigate custom model development in Pipsqueak Pro. A quick announcement, we've just started our own blog, which is really awesome. Dr. Christy Lawton is spending a lot of time writing articles for us. And this is a spot where we're releasing information, kind of about what we're doing, backgrounds on, how AI is working in biomedical research, and really where we're going from there. So this is a great technical source. You can find a lot of information about how Pipsqueak works. And oh yeah, and there's a link for it that Christy just put into the chat. So you can find it from that link. If you're on the Rewire website, you can click through to Medium from this link here, and subscribe, follow what we're doing there. So yeah, a little bit of background on Pipsqueak. Essentially, Pipsqueak AI and Pipsqueak Pro are at their heart cell counting softwares, cell counting programs. And a lot of this is really based around the idea that in different areas of biological research, or as in my case, neuroscience, our workflow often would result in some sort of microscopy or image analysis. My experiments always started with behavior. I'd be running some behavioral experiment with rats or mice. We would prepare tissue samples on slides, staining with immunohistochemistry or immunofluorescence. And then I would go image them on our confocal microscope. And so that meant that I was always bringing these, like terabytes of images back to my laptop and sitting there on ImageJ and trying to, count these cells, detect these cells. And it was, as a postdoc, it was a terrible process. It made me want to quit my postdoc. But, ultimately, we realized that this was a problem in, oops, I'm sorry, my screen popped off there. Great, Christy, can you see that again?
We can see the cartoon video.
Oh, okay. Oh, never mind. Yeah. So the problem here was that our results would often not agree between two raters, and this was undermining the reproducibility of our data. It was slowing down our analysis in the lab, and it was actually resulting in us having to redo experiments sometimes. If we weren't confident in the data that we were showing, and so. Pipsqueak was really designed out of the need to standardize that process. And so it began as an ImageJ plugin, an ImageJ macro that allowed us to sort of detect these cells with this thresholding that we were talking about just a second ago, but ultimately to standardize how the images were being quantified. Now, we've come a long way since there. And so really what Pipsqueak is continuing to offer, I think, the reason that a user wants to use Pipsqueak is that it's improving that reproducibility of the data that we're generating. But as the person that's actually using Pipsqueak, I think the analysis time is really what makes the difference. So here, our AI models are able to detect the cells in an image and produce, a quantification result within like fractions of a second. And you'll see how much faster that is even now. So, comparing this back to the like 15 to 30 minutes it would take me to analyze an image by hand in ImageJ, this is a no-brainer. It's awesome. And, ultimately that actually saves the lab money because we are doing this faster. We're repeating fewer experiments, because of sloppy analysis. And so, that's a really good win for everybody. At Rewire, we really stand behind this. We're here to help. We're always available for customer support. And so, we're pretty stoked about what Pipsqueak is doing. And so are a lot of our users. I'm not going to spend too much time here, but we first published Pipsqueak back in 2015, and it's been used all around the world. We're seeing a lot of really cool expansions of the types of research that's being done on Pipsqueak. So you can explore this a little bit further on rewireneuro.com if you're interested, but know that you're joining a big community of Pipsqueak users. And so here's kind of how things have progressed. We started off using thresholding. We were detecting, these cells just based on contrast, essentially. And that did really improve the accuracy of our data. But ultimately, Pipsqueak version 1 was a little bit clunky. The biggest problem was that it wasn't very good at detecting cells because, if you had an image like this where there's a lot of background, there's, some fiber tracks going through here, thresholding just doesn't work, especially in cases where you have like multiple cells overlapping or touching. Thresholding doesn't work. And so we took the step of developing an AI that's able to detect a lot of cell morphologies. And this was actually funded by an NIH SBIR grant. So really cool to see NIH supporting this type of thing. And so what came out of that was Pipsqueak AI. So if you've used it before, you're probably familiar with this sort of interface. This is a really great program for, walking through single, double, triple label analysis. Once you've opened up your images in Pipsqueak, you can walk through the AI process as you're seeing here, and ultimately end up, with a whole lot of detections. And this works pretty great. I think the biggest problem with Pipsqueak AI, and as we'll see here in a second, is that Fiji really limited our capability for building an interface that was functional. If you've used this before, and maybe if you've seen the video that we showed at SFN, you understand some of the problems with Fiji. If you press the cancel button, then the program would fail. If an image got moved around or closed or lost, the program would fail. And so there's some real frustrations with that, and that's what we're building around here in Pipsqueak Pro. So the last thing I'll point out here is that Pipsqueak AI enabled us to do some really cool AI customizations, and I think this is one of the differentiating features in Pipsqueak versus maybe some of the other things that are out there. We know that we haven't seen every cell type in the world, and if you come in with something that we haven't analyzed before, we have some tools that make it really easy for the AI to learn what you're looking for and become accurate for that. And so that's, I think, an important part of this process, and I'll show you how. So today, we are very excited to release Pipsqueak Pro to everyone that's here. This is a beta version. We're really looking for a little bit of feedback in terms of how this functions for you. So you might see some features improve or change over time. And I definitely recommend you check this out, but probably not publish with this quite yet. Let us really finish this for the rest of the month here, and then we'll have something that you can feel confident in. But Pipsqueak Pro is bringing a whole new look and feel to Pipsqueak, and so you can see that here. This is an interface that makes it very easy for somebody to walk through image analysis. It keeps everything within our ecosystem, and it's essentially bringing an easier workflow. Something you're going to see today is that this also improves the speed of our AI detection. So this is a really nice capability. If you've used Pipsqueak AI in the past, you might remember the period of time it takes to upload some of those images before the AI runs. And as Ace is pointing out here, one of our inspirations for building this interface was really Google Drive. And so, that's a pretty good, I think, idea of where Pipsqueak Pro is going. What we recognize is that, one of the most important parts of doing this type of image analysis is collaborating with other people in your lab or maybe across labs. And so, what we've built is essentially the ability for Pipsqueak to sync your images and your data with your Pipsqueak Pro account in the cloud, and then even to share these across other people and with other people in your lab. So Google Drive is really kind of our inspiration for that. And this is a feature that's coming out here within the next few weeks, and you'll see that become available. One of the other things that Pipsqueak is making possible is to do a lot more of your analysis right in Pipsqueak Pro. If you've used AI in the past, there's a little bit of work between finding your cells and quantifying them and then running your statistics. And I think we've recognized that one of the problems in Excel is that with large data sets, you just kind of get bogged down. Sorting isn't that easy. And so in Pipsqueak Pro, we're making it possible to walk away with group means, exactly the data that you need to plug into whatever statistical package you're using, just to, then sprint to the finish line. And then finally, we're going to enable, more co-localization features, better co-localization workflows, and faster batch processing. So today in Pipsqueak, what we've got available is single label analysis, and I'm going to show you how that works here. So we're going to head over. I've got my, my fancy wallpaper desktop, and so the first thing you'll notice is when I open Pipsqueak, all I have to do is execute the Pipsqueak icon. So that's really nice. you've got this, really clean interface here. And when I log in, I'm able to log in exactly the same way that I have been in Fiji. So this is even your same login here. So I log into this, I've got my account active, my license active. And so I can already see a bunch of the projects that I've previously been working on in Fiji, or in Pipsqueak. So similar to Pipsqueak in the past, you're able to use your license across machines. So if you're working, on your computer at work and then you go home, you can carry straight over, and this is going to be available to you. As our cloud sync feature comes online, you're going to see that all of these projects that you maybe started in the lab are going to populate automatically on your computer at home. And so that's pretty incredible for, the ability just to upload your images, get everything plugged in here and get going. You don't have to carry around your your external drive anymore to synchronize, to do your analysis elsewhere. So great. I've got, my image, I've got some of these projects that I've done before. Let me show you how we start off a new project here. So I'm going to call this John's PNN analysis on what's today's date, the 7th. So I can just pop that in there. I could even give it a little description if I wanted to. And as I create that, I'm now in my new current project. That's today's project. So I'm going to go ahead and, just upload a couple images to this. This is a really cool thing to show here. I have the ability to either upload just the images themselves, or I'm really excited to announce that I'm able to upload files and folders. And so this is really important for the type of analysis that we were doing in my postdoctoral lab with Dr. Barbara Sorb, because often our folders look like this. Maybe you can relate to it. I've got an experiment, and then maybe I have some subjects or some groups under it, and each one of those subjects or groups has a whole bunch of image files under that. So Pipsqueak is now enabling you to pull that straight up. So let's actually go over, rather than uploading this, I'm just going to show you what that looks like. So here I've got, maybe one example experiment where I've just got a couple images, and maybe I want to sort these images into subject 1 and subject 2. And when I do that, when I create these folders, I can just, drag my images right into it. And now I've got, all of those available. So let's take a look at how the processing looks. This, for example, is a co-localized image, but we're just going to look at them individually. And so here I'm going to, this is an oxidative stress marker. I can zoom in on it, really see where my cells are. I can choose the AI model that I want to run here. And all I have to do is press analyze. And voila, what we're going to see is, very quickly, those all these detections pop up right away. So similar to Pipsqueak in Fiji, one of the things that we think is really important is for you to be able to look at your cells before you accept the annotations and change anything that you don't agree with. So for example, I see one cell here that didn't get detected. I want to make sure that I add that in, or maybe there's a big cell here that I don't think is right. I can delete that straight out. So what you walk away with is an annotation set or an ROI set that you think is perfect for your image. So I'll pause there. Let's take a look at some of these questions. File types that can be imported here. Great question, Trevor. So this is even better than Pipsqueak Fiji or Pipsqueak AI. This is really going to let you upload any sort of image file type that you have. So like, JPEGs, TIFFs, PNGs, anything like that. I don't think it's going to do GIFs, but maybe in the future. And something that we're currently working on is in, in Stacks. Great question. So something that we're working on is an integration with bioformats that will enable you to export, what you like your LIF file out of Leica's software, bring that over, convert it in bioformats and open it up here. Currently, we're not looking at Stacks, but that's something that's going to come online with our co-localization capability. So, for example, I've got 4 or 3 channels and a merged image here. TIFF Stacks are something that should be available within that as well. Oh yeah, thanks, Grant. Good answer to that. Yeah, and so let's go over here and take a look at another project that I have open, because this is probably more similar to what I'm typically doing. Here I've got a ton of perineural nets. These are WFA stained, and I've got them broken into these different subjects. And so what's capable now or what's possible now in Pipsqueak Pro Beta is that I can run inference on this. I can run the AI on this image, and pulls up the cells that are detected there. And right away, let's say I agree with this. What I'm going to do is I'm going to prove the ROIs on here. And so when I do that, what you see is back here over on that image, I now have a check mark, and that's really nice because I can see that that image is done. As we enable batch processing, what you're going to be able to do is, run through all these images very quickly, produce your detections, your cell detections on here, and approve them one by one as you go. Finally, we're able to export those ROIs, and so I'll show you what that looks like here in just a second. But it's it's a really easy way to see the data come through this process pretty quickly. So yeah, Larry, really good question here. Our AI methods are extremely transparent. We really want this to be something that everyone feels comfortable with. And our approach on AI has essentially been that this platform, this AI is helping you detect the cells, but we really don't want to trust it blindly. So if you see a cell detected on here that you disagree with, that's a great thing for you to say, oh yeah, no, I don't like that one, or I do like this one here. But these are we're really utilizing a type of technology, AI technology, called object detection. And one of the reasons for that is that we think image processing is best accomplished by detecting your objects and subtracting out your background. So in this case, I'm using a rolling ball algorithm that allows me to smooth out the background signal that was in this image. And so what I'm left with is either a pure black, that's quantified as not a number, or signal, and an ROI around that allows us to really take the whole picture into perspective. If I was to look at this cell with just a mask or a segmentation around it, I'm losing a lot of information because, especially in this case, these perineural nets are not very well contained objects. They're diffuse out in the dendrites. They're hard to always define perfectly. And so I think a bounding box is the absolute right way to go with this. So we presented a lot of our methods at the Society for Neuroscience conference, and I'm happy to make that poster available to you. But I'm gonna come back to how exactly we're training our custom AI models here in a second.
There's a few more questions, John.
All right, awesome. So does it measure the whole pink area? Pixel intensities or intensities of the net edge? Great question. So first important feature to show here is that you can now make your ROIs any color you want. So boom, there you go. Now they can be blue too. But yeah, that's a really good question. So what this type of model is measuring is everything within the bounding box. The background of this image, since I've gone up to my background subtraction features, is now turned into not a number unless it's a pixel intensity that was brighter than the rolling ball determined the background to be. So this is, I think, this is what we published in 2015 and have published several times post then, because I think it's a way to take the entire cell into perspective. And as I'll show you here in our data output, what essentially we're walking away with is a comma separated file that allows us to see a number of metrics around that cell. So if you use Pipsqueak AI in the past, then sort of the data that's included in here. But most important is that we're measuring the background of that image. And then for our methodology that I've published with Dr. Sorg is, we are now reporting the mean minus background value of intensity. So that's a good way for us to, show actually signal within that ROI. A couple other metrics in here that we've gone into in other webinars, and I'd be happy to talk to you about more specifically outside of this. So yeah, let's go back to this. And do the images need to be grayscale? No, they don't. So this is going to be a little bit of a question of exactly what you're trying to quantify. So let me see if I can find one of the merged images here. Yeah, so You know, for example, I can see a bunch of nets in there. So I'm going to go over, I'm going to use this WFA model and I can run it on that and it's going to pull up those ROIs. So that's pretty nice. If you're using, you know, merged images and you want to analyze that, then that's something that's not possible. Great. And can it split channels? Yeah, that's a really good question. So Pipsqueak is set up to import the images that you've exported from your fluorescence or bright field or, you know, confocal microscope. If you are bringing in merged channels, it's not going to pull the channels out for you here. So, you know, I think the best way to approach that is actually to export each one of those channels individually, and then maybe bring over your merged channel as well. Yeah, and so, I mean, in terms of accuracy, I think the, you know, there's really two parts to that question. And so this is actually a really good segue over to the next thing that I want to discuss. Let me come back to that. And so, you know, really the accuracy of AI is going to be dependent on what the AI has been trained on. And so, you know, this is an important step in a user showing Pipsqueak what they're looking for. So for example, Pipsqueak comes, you know, ready to go out of the box with a number of custom models that, like we saw in those last images, are looking for objects that they've seen before. And so, you know, if you import your image and start analyzing it, what you might find is that AI model is not producing results that you're happy with. So here in this image on the left, you know, we see that there's a whole lot of maybe blue cells here that weren't detected. Well, our platform allows you to correct any of those issues that you see, come up with an ROI set that you're comfortable with, and begin training a custom model. And, you know, I think this is an important step to realize, because as AI becomes more and more prevalent in, you know, our world, our society, it's important to remember that it's not magic. You know, this is really just a tool that's helping us, you know, reach an endpoint. And so here, what we need to do is, you know, detect those ROIs, make those annotations, and then just press submit. And our automated system in the background is going to take those new annotations, develop a model that's specific for you, and then release it to you right through your AI model library. So this is what we talked a lot about at our poster at SFN, and I think we're presenting here again at Winter Brain, just in case anyone's going to be there. But it's a really cool method that was designed by our AI team, Will and Jacob, that's utilizing some cool experiments in the background. And so, you know, what you're going to see here over in our custom model features is, or our custom model module, is the ability to create your own model here. So I'm just going to call this, you know, test, and give it a little description. And I can associate this model with a particular project that I've started analyzing. So, you know, let's go look at, you know, one of those, or no, let's look at, you know, a project that I started the other day. And so I'm going to create this, and when I come back over here, now I can add images to that. Oh, I moved those images, so it's a little weird. But I can, you know, I can go over and I can add images to each one of these custom models, and by doing so, I'm essentially letting the AI know that these are the images that I've gone through and annotated, and that I want to train a model off them. So, you know, once I've done that and completed this part of the process, I can just start training a model, and this queues it up on that automatic server. So currently the process takes just a couple days for this experiment to run. So I press train, that gets it going. And now, you know, after that period, when I come back here, what I'm going to see is, you know, a brand new model available for me down here in my model library. So Larry, great question. Have we tested this on serial EM? We have not, but, you know, I think the biggest takeaway is that the AI should be able to detect anything that you would like to put in front of it, just with a little bit of fine tuning. And so, you know, the best way to do that is to bring it in, see if the image is something the AI is detecting currently, and then, you know, just make a couple of those additional annotations, what you're looking for, and give it you know, a place to start training. I'd be happy to work with you or anyone else individually to test out your images and see if this is going to work for that application. Yeah, great question. So, all right, well, let me come back and I'm going to talk about, one other aspect of this that I want to touch on, and that's really, where these Pipsqueak versions have gone and what's going to be available to you. So Pipsqueak is the future of, or Pipsqueak Pro is the future of what we're doing here in Pipsqueak. And so our ImageJ versions will eventually stop being supported. We're going to at least keep these up to date for the next year. Obviously, if you're running co-localization, you're going to be needing to use that here for just a little bit longer. So that will be available to you. In basic, will always continue to function. That's because it doesn't need our servers to run. If you've got it installed, it's going to work for you just fine using thresholding. So, don't worry too much about that now. But one of the biggest things that I want to point out is we've really benefited from our early supporters, our early adopters, and we want to make sure that this continues to be rewarding for you. So if you lock in your pricing now, pick up a Pipsqueak Pro license, we're going to honor that rate far into the future as things may potentially change, as long as you keep that license active. So lock in now. You can talk to Asa Brown on here or hop on our website and see how that fits into your lab's budget. But those licenses are going to make all of these versions of Pipsqueak available to you. This is just one sign-in. So if you're using Pipsqueak AI plus Pipsqueak Pro, all you have to do is sign in the right interface and it will be there for you. So we've talked a little bit about that, custom modeling, and finally, where you can find it for yourself. Christy, if you want to pop that into the chat, thank you. So just click that link. That will take you over to a hidden product page over on rewireneuro.com, where you can actually download the beta of this now. Again, this is, we're considering this a beta. I'd really love to hear any feedback that you have. And if you take the time to talk through that with either myself or Asa, I think we have a small gift for people. Is that right?
That is correct. Yeah, we will be offering a $25 gift card to anybody that's willing to hop on a meeting with me and give a little feedback, because I really do want to hear how we can best provide value to these customers. So yeah. I'll drop my email address in the chat right now so you guys can grab that as well as you get onboarded and play around with the product.
Awesome. Yeah. I mean, just to really underline that, we are a group of scientists building, products for other scientists. And so, making this something that's usable for you is really important to us. So thank you for taking a look at what we've got here. And yeah, feel free to reach out to Asa or myself. Trevor, great question. So timeline for double label on here is fairly close. We essentially are building out a module that allows these different channels to overlay co-localizations on each other. So you've used Pipsqueak Pro in the past, I know, and one of the biggest, or I'm sorry, Pipsqueak AI in the past, and one of the biggest differences you'll see is rather than being forced through a specific workflow of here's channel A, channel B, and now co-localize. What you're going to see is you'll take these annotations and specifically ask it to make comparisons between two images or two channels or three. And so we think that's a really great way for you to kind of hone in on the data sets that you're most interested in, produce those subpopulations of quantifications, and walk away a little bit faster. So timeline for this, I don't want to promise anything, but we're ultimately rolling out the full, polished version of this by the end of the year. So you should start to see these things coming online here quickly. Currently, those are all still available over in Pipsqueak Fiji. And if anyone has any specific questions about how to do co-localization in Pipsqueak Fiji, I'm happy to stay on and continue talking with you, or you can check out one of our previous webinars over here on our website. So if you're on rewireneuro.com, all you have to do is navigate over to learn more. And then we've got a whole bunch of resources here, including how to install Pipsqueak AI, the old version, which is a little more complicated than it is now, but then also some of those webinar recordings. So, feel free to peruse through this. It's great material to help put you to sleep at night. If you are, just having trouble sleeping, just listen to me drone on and that should do it for you. So, hey, Hillary, nice to see you here. Can Pipsqueak Pro still handle large images, low magnification? Yeah, really great question. That's something that we're working on. And let me kind of show you what Hillary is asking about here. I'm actually going to open up Fiji or Pipsqueak Fiji and show you kind of how that looks. So one of the things that I think we're really happy with is the way that our AI is able to look at some of these really big images. And so I don't think this person is on the call currently, but I'm gonna show you a tiled image that I came across just the other day. Let's see, where did that go? So essentially, well, we'll just look at something else here. Let's see. Great. So here we've got we've got some really big images. Hang with me here for just a second. All right, awesome. So here's a really big tiled image that came in, and this is actually already chopped down just a little bit so that we can actually see what's going on here. So I'm going to quickly run the background subtraction on this. And so what you can see is, this is a large piece of the brain. And so, what I can do in this type of image is, maybe select a model that's going to detect with, some similarity to that shape. In this case, I'm just going to try this 8-oxo model. And I'm going to come over here to my change settings, and I'm going to use a feature that we call subsectioning. And so, here I can go in and actually break this image up a little bit. And run it again, with some AI settings, and it's pretty cool because it's helping the AI really focus in, even though it's never seen an image like this before. It's never been trained on anything this big. We're able to go in, and I think it's going to produce some result here that's well a few artifacts on there, but you get the idea of how this type of approach can help the model go in and find those targets, and it's going to convert these. So I'm actually just going to close this out and walk away from it. But anyway, yeah, Hillary, that's something that's coming online, not available yet. Let's see, when we tried counting our cells, it wasn't yet. So Alexa and Kalusa, I think the best approach there is to train that AI model for you, and if it's an accuracy that is outside of that, then that might be a great time for us to hop on and chat again and see what we can do to make sure it's producing what you're expecting. Yeah, so does it require the image stack to be summed first outside of the program? Yeah, really good question. So again, over in Pipsqueak Fiji, one of the features that we enabled really early on was the ability to sum a Z stack into a 2-dimensional image. That is something that Pipsqueak Pro will enable you to do as well. But I think that's a little bit of preference in terms of how you're processing images. In the Sorg lab, that was the type of approach that we agreed on, was that we wanted to take, several microns of a Z plane and sum it together so that we were, really seeing a good relative signal across all of our images, and specifically, we were not taking a max projection. We were summing our images because we felt like that was a better approach. But that really, isn't required if you are coming in with a, one or a single Z step, or maybe an image from a fluorescence microscope, a bright field scope. You're not going to have that Z plane, and so that's totally fine. You can bring that in and analyze just a single plane, but I think for confocal, a good approach is to sum that Z stack. So yeah, awesome. Let's see, Christy, we've already talked about a lot of the features in Pipsqueak Pro, but do you want to pop up that next question there?
Yeah, hang on a sec. So yeah, we'd like to know what other topics you guys are interested in.
Anna, great question here. So the way our AI learns is something that's really important to understand. So we've created, this an enormous image set of annotations that we've trained, for example, this WFA model off of. So we have, about 10 million annotations that went into these models. As you start to customize models specific for you, your data are staying contained to your model. So this is not something that's being shared with others. And what's important about that is that somebody else's data aren't potentially corrupting your AI models. So you can feel confident in what you're seeing, that's specific and static to you. A feature that we've talked about releasing here for a while is the ability for somebody to sort of publish or deploy their model if they would like to share it with the community. I think in certain examples, for example here, Priyanka Bhushana is a graduate student that I've worked for a long time at Washington State University, and she trained up this specific 8OXO model that she felt comfortable deploying to the rest of the community. And so that's something that she can take some ownership of. It's almost like publishing her model for others to see and benefit from. And so that's something that I think we'll continue to explore and pursue as time goes on here. But currently your models are specific for you, and they're only changing. This is a very important point, is they're only changing when we click train model. And so let me go back here, since we didn't really see a great example of this. So here's just another model name. So we're going to go in and we're going to take one of the projects that I started today. Oops. And we'll create that. And so when I come in here now, I'm going to add images. And oh, sorry, I haven't saved any from that. But what we're going to see is that doesn't start training until I press train model. Now, a way that we've explored doing this down the road is, for this to be a little bit more fluid. The program may learn as you do those annotations, but this will be very apparent to you. And so when you create a static model, or when you call a model done, it's going to be done and it won't change, which is important. So wonderful. Well, I'm happy to stick around here and continue to answer any other questions that come up. I'd also be happy to run through co-localization in Pipsqueak AI if you're interested to see how that works, or even how to sum an image over there, since we've talked a lot about that. But thank you all so much for coming and joining us today. Our whole team is really excited about this release. We've put a lot of work into this. Our developers, Grant, Will, Jacob, Colton, everyone else, I think some of them are here. So they've put in a huge amount of time into developing this, and so kudos to them for building something that's really great. And I'm excited to let you play with it, and excited to show you where we go with it from here. Oh yeah, Asa, great point. Last thing to mention is to follow us on our blog and follow us on Twitter. Dr. Emily Jorgensen is here with us. She runs a lot of our social media, and she's put in a lot of great information about what Pipsqueak is doing, where we're going, and just things that we're involved in over on Twitter. So follow us there. So great. Thank you all so much for being here. It's great to see a lot of familiar faces, and I'm excited to see what you think of Pipsqueak Pro. Thank you again.
Hillary has a question.
Oh,
She had her hand up, her virtual hand.
I think she's clapping.
Oh.
I was clapping, but I do actually have a couple questions, if that's fine.
I'm happy to stick around here.
Awesome. One was for the PNNs. Have you ever heard of people using separate models, like one for really like mature, bright ones, and another for like less intense signal?
I think that's
use?
Yeah. Are you talking like within the same image, or maybe between experiments?
I was sort of thinking in the same image.
That's a really good question. Yeah, we were talking about that just the other day. Since currently you run one detection model, it comes up with your ROIs, and then it lets you change it. One of the things that we talked about was the ability to stack detections on top of each other. Especially if you were looking at maybe two different types of stains, like parvalbumin and WFA. That might be a really good way to merge those into a single image. So yeah, I like that. Like even for different types of nets that could be really beneficial.
Cool. And then the other thing I was wondering about, which I sort of just realized I never really knew, is how does Pipsqueak get the information about the like pixel size in every image, or do we have to input that manually, especially now in Pipsqueak Pro?
Yeah, so if you want to convert your pixels into, you know, micron distances or distances, whatever it might be, you're going to have to bring that conversion factor in from your microscope, because it's going to be, you know, specific for the objectives that you've got in there. It would be, you know, pretty hard to determine that, you know, outside of this. So yeah, that's something that you can still include. That is not something that's in the beta of Pro right now, but will be, you know, before the full release. And yeah, otherwise, you know, the area of an image right now is just included as pixel area.
Cool. Thanks. I'm looking forward to trying it.
Cool. Awesome. Thanks so much for coming. Great.
Hi. Sorry.
Yeah.
Hello. Yes, I was interested. What can be data output?
Great question. Yeah, I didn't spend too much time on it here because we have talked about it a little bit in one of the other webinars. You know, what you're going to see is going to become more and more usable in Pipsqueak Pro. So, you know, this is a little bit of a feature reveal here, but one of the, let's see, I'm going to go back to one of the projects that we were looking at. One of the ways that you're going to start to see data presented to you is, you know, under each one of your images, you're actually going to have access to the ROIs and, you know, the quantifications that have come from this image. So, you know, as I look at this image here, run my detection on it, I can, you know, see all of the data resulting from that right here under the image, especially after I approve it. So yeah, there it's approved. Great. Now, you know, I might want to see my results, or even potentially results from somebody else in my lab. So, you know, as the collaboration parts of the program are really important to us. You know, if Christy and I are both working on analyzing this image and we want to compare the results that we're getting, you know, I want to be able to see both my results and Christy's results. So those will be here. What we're imagining is the ability to, you know, create some group means right here from this data set, from this image set. And then when you, you know, open those up, so let's see, I'm gonna close, oops. I'm gonna just go over and find my image. How about that? So when I save those results, I can go in and I can find the comma separated value right in that folder where I was running my analysis. So yeah, I've got to figure out which image I did, but great. So here's subject 3F. I ran analysis on that image. And so, you know, Pipsqueak does a good job of keeping your images where you've stored them. It's not going to move them around. And then, you know, right next to that image, I can actually see the quantification. So this is a comma separated value, allows you to open it up. And I've got a whole bunch of different things in here. But, you know, what I mentioned earlier, I think is probably most important. And that is this mean intensity and mean minus background. These are the metrics that, you know, I've published a number of times, but we've also started reporting integrated density. And we are planning to enable the ability for users to come up with different types of quantifications that they're interested in. So if, I don't know, for example, you're interested in multiplying the, I don't know, the max by the area, then, you know, that's just kind of a function that you'd be able to write in and see in your data, see in your results.
Great, thank you.
Yeah.
John, Trevor has a question about the trial.
Awesome. Oh yeah, Trevor. So the trial is available for 2 weeks, kind of out of the box, just to put a number on it. But as you're trying the you know, beta of Pro here, I'd be more than happy to extend that as long as you need, you know, until the day it's released. So if you need, you know, the rest of December to try this out, you know, just reach out to Asa or I, and we'll keep it going for you. I can't tell you all how much we appreciate your input, you know, on this, and so I'd love to give you as much time as you need. Great. Well, yeah, one more time, there's a link to download it, so give it a go and let us know what you think. Awesome. All right. Well, thank you all. You have our email in here, and so I hope to talk to you all soon. Thanks again for coming. Oh, yeah, Trevor, for when you want to start your trial, that link will be open. It's not something you can navigate to in the menu. So if you don't see it there, that's why. But just hold on to this URL or email me if you need it, and I'll give it back to you. And that way you can find the download to download Pipsqueak Pro. But that should be available here for quite a while. So if you want to wait till after the holidays, that's probably fine.
Okay, and thank you for doing this for us.
Yeah.
Will
Thanks for coming.
the updates that you guys are implementing be automatic, or will the Pro beta form have to be downloaded again? Or
great question. Yeah, these updates come through your interface in Pipsqueak Pro automatically, but it does let you know that an update is coming and gives you the choice to take it or to hold off. Obviously, if you're in the middle of an experiment, you probably don't want to risk anything. So that option's available, but it will show up here automatically.
okay, thank you. I appreciate you guys doing this.
Yeah, thanks for coming. Hey, Shafali, how are you?
Hey, John, how are you?
Great.
It's good to see you.
Great to see you.
Actually, two questions that are related. So the first part is just, I'm a little confused. So if we have a license for Pipsqueak AI,
yeah,
we're still in trial for the Pro, or how does that work?
Yeah, exactly. So actually, that's a good question. If you go over to this link, what you're going to notice is that it's going to give you a new trial license. You don't have to use that. Go through this process just so that you get the download link to install it on your computer, and then you can log in with your same login that you currently use. And that way, since you have a yearly active license, this will continue to work for you as if there's no difference at all.
Okay. And then my other question was, if that's the case, is there going to be a conflict in having Pipsqueak AI as a plugin already or Pipsqueak Pro as well, or is that?
Nope. Yeah, that's totally fine. Since this is a standalone installable, this just ends up looking like, Excel or anything else that you have installed on your computer. So, here I've got Pipsqueak Pro or Pipsqueak AI running, additionally to Pipsqueak Pro. So yeah, no problem there at all.
All right.
I guess the one caveat to that would be if you're analyzing images from the same folder, you're going to see all of those results populate in that folder. They are named slightly differently, so you should be able to keep things clean. I mean, if you're wanting to experiment around with Pipsqueak Pro, I might just make a copy of your images so that you keep those data separate, but it's not going to overwrite anything.
Okay. Yeah, we just had like a major issue with our microscope, and so all our light settings have changed.
Oh no.
We might have to start from scratch for some of our image analysis, and so I'm thinking, should I just wait till the Pro is out, finalized, and start there, or should we go back to the AI?
Good question. Yeah, maybe just hitch back with me when you get the microscope up and going, and I'll give you an idea.
It's up and going right now. It's just a labor shortage of getting someone to actually sit there and take the images.
Yeah.
With the new settings. So we need to make a decision soon. And so I wanted to, ask, this is with Lee over at UH. And so I wanted to see if it's worthwhile waiting for Pro and just starting with that.
I see. Yeah. I mean, if you feel comfortable holding off for, maybe a few weeks or the rest of the month, then Pro is probably a good way to go, especially if you only need single label right now. But if you want to do double label,
Right.
might anticipate going back to AI just for a little bit longer.
Oh, okay. Yeah, no, it's single label right now, I think.
Okay. Yeah.
Thanks, John.
Yeah, absolutely. We're in the process of doing some really deep comparisons in the quantification that's coming from both of these programs. what I anticipate is that they are going to be identical, statistically not significantly different, but I would encourage, your publication data set to all come from one program or the other.
Oh, yeah, of course.
Yeah. Okay, great.
Definitely. Thank you.
Yeah, great to see you.
You too. Good luck.
Thanks.
You guys are doing great, so I'm sure that it'll only improve.
Thank you.
We've made a lot of progress just in the last few months.
Yeah, it's cool to look back just a year ago and kind of see where we were. Our whole platform has come so far. So again, thank you to everyone that's been an early adopter and given us feedback.
Well, any other
Great.
questions? There's a couple people still here.
Cool. All right. Well, let's call it there. And if you have any other questions come up, feel free to reach out to one of us.
All right. Stop the recording.